All right, let's jump right in. We're going to tackle a huge question today. Why is it that some parts of our body can heal up almost perfectly, but an injury to our nerves, that can last a lifetime? So, what gives? Let's get into it.
I mean, think about it, right? You get a cut on your finger and it just heals. No big deal. But a serious injury to, say, your spinal cord, that can be completely life-changing, permanent. So why? What's the fundamental difference here? Well, it all boils down to how our nervous tissue reacts when it gets damaged. Okay, so let's set the scene. We're starting right at the moment of impact. Picture this. One of your nerve cells has this super long wire, it's called an axon, that it uses to communicate. Now, imagine that wire gets severed. That single event, it sets off this massive, cascading chain reaction throughout your body. And right away, we hit our first major clue. You know how skin cells are always making new copies of themselves? Well, neurons don't do that. Most of them are what scientists call postmitotic, which is really just a fancy way of saying they've lost the ability to divide. So once a neuron dies, that's it. It's gone for good. This is huge because it means the body has to be able to repair a damaged neuron. It can't just make a new one. The stakes are incredibly high from the get-go. So that axon gets cut, and boom, the neuron's main headquarters, the cell body, gets an emergency SOS. It basically drops everything it's doing, all that normal data-game message sending, and switches into full-on crisis mode. Its one and only mission now, survival and repair. And this emergency mode, it actually has a name. It's called chromatolysis. The easiest way to wrap your head around this is to think of the neuron like a factory. Inside, you've got something called Nissl substance. That's basically all the machinery for making proteins. When the SOS comes in, the cell completely reorganizes its entire factory floor. It's retooling everything just to pump out a ton of new proteins, all the spare parts it needs to rebuild. It's a true all-hands-on-deck situation. And what's so cool is that you can literally see this happening under a microscope. The whole cell body starts to swell up and get round. The nucleus, the brain of the cell, gets shoved over to the side. And that factory machinery, the Nissl bodies, these start to fade. Now, you might think this looks bad, like the cell is dying, but it's the total opposite. It's the physical evidence that the cell is fighting for its life, retooling for this gigantic repair job. Okay, but what about the other piece? What happens to the part of the axon that got cut off from the main cell body? Well, it's basically an orphan now. It's completely disconnected from its power source and its protein factory. There's just no way it can survive on its own. This brings us to a process called Wallerian degeneration. Because that orphaned piece of the axon is cut off from the factory, right? It's not getting any new supplies, no new proteins, so it just starts to fall apart, to disintegrate. And before the body can even think about rebuilding, it's got to clean up the mess. Time to send in the cleanup crew. And this whole cleanup happens in a few key steps. First, the axon itself starts to break down. Then, that protective insulation around it, the myelin sheath, it shatters into little fragments. And finally, the cavalry arrives. Specialized immune cells swarm the area and basically eat all the debris. They phagocytize it. This cleanup job is absolutely critical for any repair to even have a chance. Okay, so the site is cleaned up. And this is where the story completely splits in two. The chances for recovery, for actual healing, depend entirely on where this all happened. Was it in the peripheral nervous system, the nerves in your arms and legs? Or was it in the central nervous system, the brain and the spinal cord? Because the outcome is going to be wildly different. So let's look at the peripheral nervous system first. In the PNS, we have a hero, and it's called the Schwann cell. After the cleanup crew clears out the old, broken axon, these Schwann cells don't just hang around. They start multiplying like crazy. They line themselves up and form this incredible guiding tunnel, basically creating a perfect pathway for the new axon sprout to follow. It's like they're literally rolling out a red carpet saying, "Hey, new axon, grow this way." But in the central nervous system, the story is completely different. It's kind of a tragedy, actually. There are no Schwann cells to be the hero. Instead, a different type of cell, called an astrocyte, rushes to the scene. But instead of making a nice, neat path, they do the opposite. They form this thick, tangled mess called a glial scar. And this scar acts like a giant roadblock, both physically and chemically, that just stops any new nerve growth cold. So let's just break it down. In the PNS, Schwann cells make a clear, guided path. So repair? It's possible. Good, even. But in the CNS, you get no path, just a big, ugly scar that blocks everything. So repair fails. And that, right there, is the fundamental reason why a paper cut on your finger heals, but a spinal cord injury doesn't. All right, so we've got our helpful path makers and our destructive roadblocks. But what is it that actually tells the new axon sprout to start growing in the first place? What's the go signal? Well, that's where the chemical messengers come in. You can think of them as the conductors of this entire healing orchestra.
These messengers are called trophic factors, and what they are these tiny little proteins. The target cell, the muscle or gland the nerve is trying to reconnect with, sends them out. It's like leaving a trail of chemical breadcrumbs. These breadcrumbs don't just say, hey, come this way, they also provide all the nourishment and support the growing axon needs to survive that long journey back.
And there's a whole family of them, with names you might have heard of like nerve growth factor, NGF for short, or brain-derived neurotrophic factor, that's BDNF. Each one has a very specific job, kind of like a key for a specific lock, supporting different kinds of neurons. So whether these signals are even there or not, that's another massive piece of this puzzle. Okay, let's do a quick recap of the whole process. When there's an injury, the main cell body goes into emergency mode, that's chromatolysis. The broken off part of the axon gets cleaned up, that's Wallerian degeneration. In the CNS, astrocytes jump in and form a scar. And all of this leads us to that one big conclusion. Repair in the PNS? Pretty good. Repair in the CNS? Really, really poor.
So if you're going to remember just one sentence from all of this, let it be this one. A nerve injury triggers chromatolysis, Wallerian degeneration, and gliosis. The end result is that regeneration is very limited, and it's always going to be better in the PNS than it is in the CNS. That's really the whole story right there.
And that, of course, leaves us with the biggest question of all, the one that scientists are racing to answer. Now that we understand the good guys, the Schwann cells and the growth signals, and the bad guys, those glial scars, can we do something about it? Could we find a way to mimic what happens in the PNS and apply it to the CNS? Could we one day actually teach the brain and the spinal cord how to heal themselves? I mean, finding the answer to that would change everything. Thanks for joining me.